Thank you for coming. Today we have Sara. Finally, after moving her seminar several times, also a bit about her work. She's a PhD student. She started in January this year, and she's been working with Marco and Gila. Okay, you can go.
Okay, so thank you everyone for coming. You didn't need to come, but thank you. So I'm gonna present Micromonas, which I will introduce later, but it's a microalga, really tiny, and then maybe some other micro things we shall see. So when we think about the ocean, most of us think about this big blue, impressive and imposing blue mass, and with all these big animals, with the fish, the whale, and all these things. But I want you to change the perspective to the micro scale, in which there are also really funny and really nice forms, like diatoms, phytoplankton, zooplankton, and they are actually nicer than the big animals. But first, I want you to look back at what we probably learned from school, or at uni at least, which was this linear chain, which is the so-called marine food web. And it starts with the primary producers, which is basically green things. And then it goes up through zooplankton to fish, and then to the big animals and all these fish that we shouldn't eat because they have heavy metals. But this vision has changed a little bit, because in 1983, the role of the microbes was actually rediscovered. So it was coined the microbial loop. And actually, there is a flow of matter within the microbes, which actually is impacting the whole structure of the ecosystem. And also the viral chain, which is also a big contributor of the organic matter within the ocean, which actually allows for the structure and the lives of other big organisms. So here you might see like loads of arrows and loads of things. But the most important here are the primary producers, which is the phytoplankton, which are strictly photoautotrophs. So that means that they eat the sun. So they create organic matter from the sunlight. And then we have mixotrophs, which can do both. They can feed on light and also feed on other organisms. And then the peak of phytoplankton, which is phytoplankton, but smaller. And then on the other side, we have other producers or other contributors to the flow of organic matter, which could be bacteria, archaea, and viruses and parasites, because they also release organic matter that goes into upper layers of the network, let's say. So that was the connection between organic matter. And now there is another kind of connection that is not always thought when we think about the microscale, which are physical connection, because these animals, some of them, for example, some diatoms, need to be together, let's say, because they do sexual reproduction. So they need to encounter at some point, and then they also need to look for some food in some cases. So I would like to speak now about motility in phytoplankton. So most of them have flagella. There are other strategies, so they can glide, they can segregate mucus, but I want you to think about flagella today. And I'm going to speak about Micromonas, which is a chlorophyte, and it has one flagella, as we can see in here. It's really tiny. It's 1-3 microns long, and then the flagella is more or less 1 micron. So this is the typical flagellum structure, which maybe someone has already exposed in these seminars. So the typical flagellum is just a basal body, transition, the cilium middle, and the ciliary tip. And in organisms like Micromonas, which is another organism, it is quite well understood how it's assembled. So they know all the proteins in each of the areas. And what happens with Micromonas is that also due to its tiny size, it is not well known what is the composition or the structure. And also this is on another topic, but it's not well known how this flagella is divided. So they don't know if the cell, in the same cell, there are 2 flagella growing and then divide, or the flagella is something that creates a posteriori. So with this in mind, we wanted to know how was the assembling and the composition of the flagella in Micromonas. So we went up to the Institut Pasteur in Paris, And we try to stain our cells with actually they were antibodies for human cells, because this group is working with trypanosomas, which is the sleep sickness. So we tried the antibodies that they had to see if we were able to see some bits of this staining and how they were distributed along the flagella. But as you can see, the stainings are not very well defined, so we should actually repeat the experiments because it's so tiny, and the way you manage the cells, it's really delicate because the micron here, the scale you cannot see, but it's most of them is like 500 nanometers. So actually it's really complicated. So we also saw them under the electron microscopy, and we couldn't get nice images for the same reason, because the cells were broken when we fixed them. But we could see some twists on the flagella, and then some other bits that are still not known. And then these Hayley points are called, which are not, they have been reported before, but we don't know what are they for. So we have inconclusive results in this part, and we look forward to maybe retry experiments and see if we can get something from this. And then, okay, they have a flagella, so it's for moving. So let's go to what's happening in reality, no? So Marco showed these videos at some point, and what happened, they were in the lab, and one student of him just recorded these bats in the lab. So he was recording for 2 hours for Micromonas, and then they realized that at some point one cell would lyse, would explode, and then it would create this halo movement surrounding it. So now this is interesting because it's the first time that it was reported. And we thought, okay, what's happening in the natural environment? Because this alga is infected by loads of viruses. Some of them are virus specific, and then there are other viruses that are also infecting other phytoplankton. So we were thinking, like, what happens when a cell is lysed, but by a virus and not spontaneously? So then we saw that there are no official records for it. So now we set this as an objective, like, let's see what happens. Let's record, have a proof, let's say, of what happens when this infection is happening. How is the dynamics? So we contact Corina Broussard, which is from the Netherlands, and he will send us some viruses which are on their way. So since we still don't have any viruses, but we do have Micromonas, we started some kind of preliminary experiments with Micromonas, which is what we have. And then we thought, well, this kind of effect when the allo is forming, it might be due because there are some infochemical that is kind of like warning the cell to move away from the live cell. So we thought, okay, so maybe this is a chemotaxis response to the lysate of the virus or of the cell. So then now we are trying to do these experiments, which is just like a capillary, which is broken, as you can see, but it's a capillary which has been previously filled with the cell's lysate. So all the compounds from the cell is within the capillary, and then surrounding them is just a sample of normal cells which are moving. This is a 4 hours experiment, so you are just seeing like images every 15 minutes. So our hypothesis would be that they do negative phototaxis, actually, no? That they would go away from the lysates. But we still are working on it. And we are hoping that when we get the viruses, we are able to test what's happening. So why are we doing this, no? So Micromonas is actually very important in the Antarctic water, and it's been seen that climate change is warming and acidifying the water. So all the composition is expected to change. And also if you remember going back to the microbial loop and the variations and all these diagrams, it's really important the position of the phytoplankton and the virus and zooplankton. So the grazings are basically zooplankton. So it could actually invert or reduce the amount of phytoplankton the primary produces, and that could impact the whole structure in the whole ecosystem. Sorry. So the future work, we will divide it into 2 points, and one would be at the population level. And it's basically, once we have the viruses, we want to see what's actually happening with the viruses. And then we know how this expansion, this spread of these cells is happening when they are all alone. So we want to know what happens if you just have one cell infected by a virus, how this process, like how this infection is spread through. And then also, coming from a physics route, we also want to see what is the background currents, like is that also impacting the movement of the cells? And then, as we already said, is there any infochemical? Like, is it because you would expect a different composition within the cell when it's been lysed spontaneously than when it's been lysed by a virus. So we want to see and check if there exists any infochemical. It might be difficult because we speak about really, really small molecules. And then at the single cell level, we will develop some different microfluidic devices and maybe other optical approaches. And then it's kind of the same, but reduced to a single cell scale. So we want to see what is the motility of one only cell, like how is it moving? Like, is it moving like a bacteria that has the run and tumble, or how is it? And then how does the composition of the cell changes in a single cell, which can be done through quantitative contrast microscopy, which is really nice because you can see the kind of like the changes of one only cell within the, well, when it's being infected. Like we know that the chloroplast, for example, is the last bit that is infected because they actually need the cell to be kind of like alive so that the virus can live. And then the last point would be like, what is the rate between viral cells and algal cells? Like, do we need a minimum of viral cells in order to infect one algal cell? Because that could also impact what we said about the ecological importance. And this is it. Thank you. Any questions?
I have a macro question.
Maybe I am not able to answer.
So in nature, do they bloom?
Yeah, and that's why the viruses are that important because they are the ones regulating the abundance. Yeah, they do bloom in the Antarctic and Arctic. Yeah.
And the densities, so because I'm thinking that in order for this mechanism to be relevant, the average distance between the cells needs to be really small because they are very small. So the signal will diffuse very quickly and we won't see it. So have you checked that the densities in the field are comparable to the densities in this kind of experiment in the lab?
I haven't checked. No.
Because I think that could be a fairly easy calculation that you could do. No? How
Yeah, I
would
think
this-
people have calculated the approximate number, well, the abundance. But the thing is that most of people also work with metagenomics and genomics because it's the easiest way to quantify kind of like the number of cells. So I think that Corina actually has done this, but I don't know now, but I will do it. Thank you.
Well, one, if the group is going to Antarctica to measure the density, I can.
Okay. Okay, I also want to
Yeah. On the bars, because I think they are very quick, no? So is it calculated if the scale for the diffusing plume? So are cells reacting to a diffusing signal? Like the first cell might be acting to a diffusing signal, but is the time scale of diffusion faster or slower than the time of reaction of the other? So because if it's slower, then it means that it's a collective behavior. So the cells are reacting to a behavioral answer of the other cell and not to a chemical. So the first cell answers to a chemical signal, but the other cell reacts to the behavioral answer of the other cells. So
Yeah.
have you,
I mean, that would be one of
the
the things
scale
we-
of diffusion or whatever is inside the cell, is it slower or faster than the cell of the scale of reaction?
Rates are big, big. I think,
Okay,
I
we
think
can
it's
talk about it
in
later.
his student thesis. I don't know if we measure this.
Yeah.
We had a look the other day together, you should know.
Okay.
We had a look at his thesis the other day. I don't know.
It's diffusion.
It's diffusion. So it's fast. Diffusion is faster than okay.
We can talk more about that.
Okay. Should we?
I wanted to ask about this experiment that you are doing with the capillary.
Mhm.
Can you explain it more? Is it that you put inside what the cells spread when they die? Is that?
Yeah, well, I am optimizing this system, but that it's like you have just a cover, well, slide, a glass slide, and then you do kind of like a round or a circle with Vaseline in this case, and then you can just fill the capillary before. You put a drop on the slide, and then
But how do you get this?
the
The
lysate.
Yeah.
Yeah, I'm using the ultrasonicator. So I first have to
Kill
centrifuge all the cells.
cells.
Yeah.
Ah,
basically
okay.
clean them.
So you
Yes,
kill the cells and then you put them inside the capillary
yes,
and then you throw this.
yes.
And have you seen any response?
Not yet, because I've been optimizing the experimental bit because I had some currents on drift, so that was not. I mean, I cannot see if there is a real response if the background is moving. So I need to enclose it properly so that there is no leakage.
Okay, because you have currents inside.
Yeah,
this.
yeah, because it was not properly sealed, but I think we can find the way of
Okay.
doing it. Yeah.
Maybe that question about the unanswered questions on the vaginal structure.
Okay.
I didn't understand really what's the constraint. So it hasn't been answered. What's the constraint? Is it a spatial constraint too small, or is it also time scale constraint? Because you said the division, we don't know if it's this division or not, it's too fast, or what's the constraint?
Well, the thing is that it's too tiny to actually observe it. Like there are papers in which they have fixed the sample to observe it under the electron microscopy, and then you can see a cell dividing, but it does this just lag, let's say. So you are not, well, I don't think you can get, I mean, you cannot see when the cell is dividing. So it's also, I guess you can look at the circadian rhythm and see when they are meant to divide, like in other organisms, they divide after dawn, for example,
The what?
after dawn. So I don't know if in here that has been done. But I mean, we do know how it divides, but we don't know what's happened with the flagellum.
Well,
What are you doing with the antibodies? I mean, what?
So the antibodies, they actually bind to X proteins. So you use them if you have your cell under the microscope and you illuminate the cell and you have some signal, that means that has attached to somewhere. So what we wanted to see is that they were specifically attaching to different bits of the flagella. So if you have your flagella, here we have the basal body, so maybe the antibody was here, or the intraflagellar transport, for example, it would be like at the tip. And then you can make kind of like a map of what are the proteins within the
You try
flagella.
different antibodies because you can map to
see
which
where they
protein
are.
each antibody. I mean, you can know the kind of protein from the antibody.
Exactly, like
Sí.
you might not need this, but there is one antibody that binds to alpha tubulin, for example, or another one to beta tubulin, and then you can,
It's the thing in
well,
biology.
or you know, at least in another organisms, what they
Okay.
do. Yeah.
Okay.
No questions? Okay, we can leave it here. Thank you, Sara.